is the second seminar of the Open Research Meeting, Community Meeting seminars. These seminars are organized by the International Committee on Open Phytolith Science. There will be monthly online meetings on different open research topics with time for a presentation and hopefully a lot more time for discussions and exchange of ideas and tips amongst ourselves. So if you're more than welcome to suggest a topic you want to hear and learn about during these seminars. We have some planned already that I'm going to show you in a second, but we're very open to any suggestion, both in terms of topic and in terms of speaker. So if you have anyone that you're interested in inviting, in us inviting, just let us know and we will gladly do it. So today, as I was saying, it's the second meeting. We have with us Rosalie Hermans, who is a PhD researcher at Vrije Universiteit Brussel. And I'm Carla Lancelotti from Universitat Pompeu Fabra in Barcelona, committee member of ICOPS, and I'm hosting today's seminar. Before giving the floor to Rosalie, which is actually the person we are here to listen to and to hear about, I just wanted to remind you that we have a code of conduct during these meetings. It's nothing unusual, but just please try to be kind and respectful when you address any the speaker or any of your fellow participants to the seminars, and obviously we won't tolerate any abusive behavior. As I was saying, these are monthly meetings, so the next seminar is actually in a bit less than a month. So in May, we're going to have two seminars. The next is on the 30th of May. It's again a Friday. It will be at the same time and in the same format, and we'll have Dr. Francesca D'Agostini speaking about starting your career on open publishing, or well, starting your road on open publishing. And then the next one will be on the 27th of June. The speaker is yet to be confirmed, but we have a lineup of very interesting speakers, and I'm sure that it will be someone very interesting to listen to. On the screen here, you can see our contacts and our resources. So we have a website with all the information about what we are doing, both future and past. So if you've missed some of these seminars, you can always have a look at the recording of the past seminar, both for this seminar series and as well as previous activities. And with that, I really stop and I'll stop sharing and stop talking, and I'll give the floor to Rosalie, who's gonna explain to us her work on the creation of a online digital images collection.
Thank you, Carla. Okay. Okay. Can you see the screen?
Yes.
Okay, perfect. So, hi everyone. Thank you also for inviting me to talk about my work. It's really nice to be able to do that, and also nice to have all the fellow phytolith researchers together in a meeting. To start off, I would like to tell a little bit about myself. So I'm a PhD researcher in archaeology at the Vrije Universiteit Brussel in Belgium. I'm now in the final year of my PhD, and in my PhD, I actually worked a lot with modern plant material because the aim was to develop these reference collections and to work on modern material to be able to apply it then in archaeological contexts in Northwestern Europe. But I went a little bit down the rabbit hole, and I ended up spending a lot of my PhD working with these phytoliths that are plant-based. And so on the one hand, I did a lot of reference collection work, and on the other hand, I did a lot of morphometric work on modern phytoliths as well. And on the left, you can see me, where a colleague noticed that my skirt was in the same color as my phytoliths that I was doing morphometrics on. And so the part that I will be talking about today is actually the part where I developed a modern reference collection focusing on dicot phytoliths in my PhD. And that turned into a paper a couple of months ago. And so we published this paper, which is presenting a modern plant-based phytolith reference collection, focusing on phytoliths from dicot taxa. So this includes shrubs and trees and herbs who are occurring in Northwestern Europe. As part of this work, and this was also incentivized by the work of ICOPS, we made a curated phytolith image data set, which is publicly available to the phytolith research community. So it is around, I think, 1,964 labeled micrographs. And with labeled, I mean that for every image, there's taxonomic metadata attached to it, together with the morphotypes that are on these pictures. And so they are in a repository. And in this talk, I will walk you through the workflow that we use to create and share this data set, also highlighting a little bit the benefits and the challenges that I personally endured during all this work. And then I'll also reflect a little bit on how this initiative fits into the broader landscape of phytolith image sharing, and also sharing some of my own questions and thoughts of what we should do with these data sets. And I really hope that we can use the second part of this presentation for some discussion, or I've been working on it for several months, and so it would be really nice to really hear what other people are thinking about this work and what we should do with it in the future. So the actual workflow, I will go over it step by step. So for every step, I'll briefly outline what we did and share a few practical tips. Some of these tips are very straightforward, but this whole pipeline of actually taking images and putting them into a repository for phytolith research is something that was rather new for me, and I also didn't find that much resources for it. So therefore, I think it couldn't harm to actually overshare. And then I'm also very interested from your side on if there could be improvements or what might be added, or just along all these steps. So a first part, of course, first step is actually take the images. This is actually something that when I process all my modern plant material, the first thing that I do at a microscope is look at my samples, and not doing the counts yet, but really want to get to know my samples, what's in there, what does it look like, are there things in there that I don't recognize? And during this first step, I already started taking pictures. And I think that's quite important to do, is that you should really integrate taking pictures if you find it important to do, and if you want to integrate in the workflow, to do it from the beginning. Then I also use those images to actually think about what I'd seen, and to actually discuss it with other people. So that's my first step, I would say, is integrate it while you're doing the microscopy work, and don't just add it as a step in the end. So in this step, it also requires quite some organization. So for file naming, I decided to be consistent in how I would name things. So for me, I decided to go with some taxonomic information, including the family, genus, species, then a lab code, the chemical processing method, because some things I process with dry oxidation and others with like a combination of dry oxidation and wet oxidation. The herbarium, the magnification from microscope, the image sequence number, the herbarium where the specimen came from, my funding agency, and then my own name. So for me, this was sort of like a basic information to have in the file name, so I could easily organize things. And because I did this work while I was in different places, I also used different microscopes to do it. So I think keeping it like organized is a very important step. And yeah, also including adding the scale bars, because I know you can do it afterwards, but then you do have to know where pictures came from and magnification and so on. So I actually like to do it immediately. And then another tip I would give for this step is that if you are comfortable with digital tools, I think establishing a uniform file name is quite manageable. If you have a well-organized sample table, you can automate or batch edit file names. For example, if you want to change species names in file names or do whatever, I would not recommend to do it like file per file if you have like large data sets. Yeah. Second step, I decided to use Adobe Lightroom to do this. So I imported all these images, and the aim of this step was actually to assign the metadata, which is in my case, I really focused on assigning morphotypes to the images. So sometimes in one image you can have several morphotypes, and what I would do then is assign the morphotype codes as keywords, and you can add several keywords to an image. And then I also added some other information, which is the species name, the plant part, and the lab code. And of course, this is a step where you don't always have morphotype codes for everything yet, because there was at this step also still some morphotypes that hadn't been named before. So then I would just say to check, or I have to come back to this. And this was kind of interesting in the workflow as well, because I would then group all the things that I didn't know or that I still had issues, and used this image data set then as a tool to have discussion, because I did lots of my identifications together with Professor Caroline Stromberg, who is based in the US. And by the time I was doing these identifications, I was based in Belgium. And so we would actually have online meetings to discuss all this material. And then an image data set is not perfect, because it's not that you're spending time on a microscope together, but it is definitely helpful. And you can have very fruitful discussions. So for me, the image data set at this step was also something that helped me in my training for identifications. And in terms of using Lightroom, I would say it's very user-friendly. You can also easily filter on keywords to sort of see one morphotype grouped together in all these images and see if it makes sense what you have been grouping as one morphotype. You can also easily batch rename these keywords. I think the only downside is that it's not free. And therefore, of course, it's not accessible to all of us. So maybe there's also alternative softwares. It may be something you can also just do on the computer. But yeah, I really like the user-friendly interface. So this is then also the step where with these morphotypes, where we had to decide what we were going to give new names or not. And so the International Phytolith Committee for Taxonomy, the ICPT, is currently working on nomenclature names for dicot phytoliths, so for all these morphotypes. So I didn't design these new names for this paper to not interfere with their work. And so what we did is we actually used the nomenclature system from the dissertation of Caroline Stromberg. And then what was not in the dissertation, that's what we actually gave, what we would newly describe. So here you see in yellow some of these newly described morphotypes. And in order to do that, we did follow the ICPN 2.0 guidelines to do that. So, but yeah, this is also that we did based on discussion from all these images and how we would name it. And yeah. In the paper, there is then a supplementary data file that also includes all these descriptions and images of these newly described morphotypes. So this is something that this information is linked to, can be found in the paper, but is not linked to the metadata of the images. So the metadata only has the morphotype code. And in order to have like the information on the morphotype, you do need to go to the paper. So in the next step, I would export the data, so to get it away from Adobe Lightroom. And in this step, yeah, I think the important parts is like deciding what kind of the metadata that you want to attach to it. So in my case, would be just like a title that included species name, lab code, and the plant part, and then the keywords to do that. And I also added a watermark on the images here. And then something to consider in this step is your file type that you're going to use to export it, because many of my file types were TIFF, which is they're quite big. And as you can see in the next slide, so around 1,000 images, they ended up being 70 gigabytes in volume, which I think is already a lot. But it is also quite feasible considering it's 1,000 images. But if it would have been TIFFs, it would be way bigger. So I think something to consider here, if you want to share it with other people, is that you are considering a file format that's not too big. But I know then you're also losing some information. So yeah, something to discuss. And then in the last step, I would put them in an online repository. I decided to use Zenodo, which is a free open access repository. And you actually get the DOI assignment for the data set that you can easily add to the paper. And previous work from ICOPS has also like these nice guidelines on how you can use these repositories, on how you can use Zenodo. And then there's also some these Open Phytolith publishing guidelines on what is expected if you are publishing a Phytolith paper, on what needs to be in there in terms of methods and results on how you can do it. So for this part, I'm not going to go too much into it, but I do want to say that the ICOPS has these nice resources to actually help you do this. So then it brings us to the experience that I had, why I think sharing Phytolith images matters throughout the process. And we go over them step by step. So I think the first part and it aligns with the FAIR data principles, is the reproducibility and the transparency of the data. So for all the species that I described in this paper, I also did a literature search to see if other people had already been investigating phytoliths from these species or from the genera or from family, and to see if I could find something that was similar in order to also get some information on the diagnostic character of the phytoliths. And then you see that some people in their work are sharing these very nice descriptions or really good pictures, but sometimes there can be like these differences in small things. And here I have this example that actually illustrates that. So what I found in the Asteraceae family were these articulated trichomes, but they had like this striate surface. I don't know if you see my pointer when I'm presenting, but it's like these arrows here. They actually show that these striations, the ridges, they are oriented like this. While when I was reading in literature, and people were also reporting on these articulated trichomes that would also have striations, but then you don't know what the orientation of the striation is. And then if you get an image, then you can see it for yourself, and you also know, okay, this is similar, this is not similar. And so a lot of parts in the paper so these researchers have been describing these phytoliths, but we are not really sure if it's the same thing. And I think in that regard, an image data set together with descriptions can definitely help to resolve these problems. The second one is, I think the sharing images really supports identification skills and training, and that goes in both directions. So for example, for my own training in my PhD, having these discussions with other Phytolith researchers about the images was very helpful in my own training process of being able to identify phytoliths, but I think it also goes the other way around, that there is a data set for people that are not so familiar with phytoliths and who are still training. It is available, it is out there, people can actually use it. And since that it has been online, I actually already received some emails from other researchers saying like, oh, we've been having a look at your data set. Is like, this is what we saw for that certain plant family. This is what we are seeing in our sample. So what do you think about it? And then I think you have this sort of back and forth process where you all learn from it. And I especially think for the dicot phytoliths, which is an interesting group of phytolith, I would say, because all the dicot phytoliths look the same until they don't. It's super important to actually visually support everything you find. And a nice example of that is in the stomata, the dicot type of stomata, which is a very redundant morphotype because you see it in many samples. And normally it looks like a traditional stomata type is the one you see on the right, where you see the two guard cells, with a sort of separation in between. And then in prickly poppy, we saw these, what we think is a dicot stomata, but we're not super sure. Having this fusiform shape, it also doesn't have like the partitions, like the clear guard cells. But yeah, this doesn't look the same. So in that regard, an image also helps because you can tell like, okay, this is what in the end we did with this morphotype. So we ended up classifying this as a dicot stomata, but it does look a little bit different. And for the dicots, that is for many things, like for the epidermal cells as well. They can be so different, but the differences are very subtle. So, and then the same thing goes for just phytoliths that we don't know. So there's still, this is, for example, a type of phytoliths. There's one who is trilateral, another one bilateral. I also don't know if they belong together. They come from the same sample, which is a leaf sample of black mustard. I don't know what it is. Caroline also didn't know what it was. I also asked some other people, all didn't know what it was, did some literature search, also couldn't find anything. And then I'm also quite happy that the data set is out there, because at some point, maybe someone will also contact me and saying like, hey, I've also seen this, or I think it's this, or, and yeah, so I think you're definitely building towards something with other researchers as well. So this is like a question mark still, what it is. And then I also think sharing images, especially in phytolith research, it encourages dialogue and trust. So on this graph, I actually put a table, a figure that comes from a paper from a colleague of mine, and she's not in the archaeobotany field at all. So she's an osteologist working with cremated remains, and she's trying to do these sex estimations based on these cremated remains, which is apparently a very tricky field because there's lots of methods, there's a lot of uncertainty, different labs do it in different ways. And so she did this questionnaire asking different labs and different people, do you trust your own sex estimations? And do you trust the sex estimations of other researchers? And then basically from this questionnaire, she found that people are not comfortable in their own identifications, and they're also not comfortable in other people's identifications. And I think this is quite striking because for phytolith research, it's also this thing where you're spending a lot of time alone at the microscope. It's not always easy to get feedback based on the things that you've seen. And yeah, it's also the same thing if I'm sometimes seeing phytolith images in a paper online, then I'm also not always, I would not always have done the same classifications, I think, but I also don't know because I didn't see the material myself. So I think the phytolith field is definitely a field where there's a lot of room for discussion. And I think there we also have to use all our tools that we have in order to encourage dialogue and trust, to be more comfortable in our own identifications and to be also more comfortable in other people, their identifications. And then we come to some challenges of sharing phytolith images. And these are all not new things. So I think definitely in previous ICOPS talks, some of these things have been discussed. The first one is, of course, the two-dimensional limitation. And I don't think we will ever go over the fact that it is 2D and it will never reach the same quality as what you see with your own eyes under a microscope. I did want to say that we do now have some methods that are allowing us to go a little bit more into 3D, like on the computer as well, with this special type of microscope. So you have these slide scanning microscopes, and this is a microscope that can basically scan a whole microscope slide. And then on the computer, you can like scroll down and up to actually go through this and go left and right, up and down to actually go through this slide. But I've done this once and it's very time consuming. It's not very easy to do. It's definitely not the same thing as actually having a microscope slide. And then there's this the confocal images as well. So they come from a microscope that can actually image a phytolith in what we call a Z-stack. So it's basically taking images whole throughout the phytolith in different fields of depth as well. And so here are two examples of confocal images. So on the left is one and fluorescent images of an elongated dendritic phytolith. It's another part of my PhD where I use these images to 3D model the phytoliths. And then on the right is a palm phytolith that was also taken with a confocal images, a confocal microscope part of a paper from 2024. And then the next thing, I think, yeah, the contextual information is crucial. So if you're sharing images, it's definitely a visual guideline, but there's still a lot of information that needs to be attached to the images. So taxonomic information, where the sample came from, and something that I also did in our paper is adding images of the plant tissues that were processed, because I also think it's nice to have a visual guidance of what was processed. Like if the phytoliths, if for example you have like these very large size phytoliths, you might also wonder, does it come from like a large plant tissue, something like this? And I actually like to have this type of information, and therefore I also added these images of the actual samples to the paper. But yeah, then there's so much more information you can add as well. Sample preparation method, imaging details, the morphotype classification, like also the same thing like with this stomata type that we just saw, like with these images, the only metadata that I added was that it was then this ST1 morphotype, but I didn't add that it had like a fusiform shape, for example. So in that regard, I definitely think that we could add more information to the images, but I also wonder where the cutoff is. And now all this information is in the paper, so they go together. But it would also be more nice to have a more like standalone thing that you don't need to have all these types of documents in order to make something out of it. And then the interpretation notes as well. So what you see, how do you make an interpretation of an image? Yeah. So isn't a phytolith type that you often see? Is it diagnostic? And yeah. And then another issue, and it's also definitely going to be an issue for this data set, is the ontology. So it's basically the nomenclature. And because ICPT is working on these new names, that means that in future, the names are going to change. So many of the names in the paper are going to change. And so, for example, this is the table from the paper. You can see that there's like two types of trachearies that Carla described that are being merged in what we have in the ICPT from 2019 as tracheary, annulate, or helical. And it's a good thing if two categories merge into one, but then I also wonder what happens if you have one category that has to be split into like a new morphotype system. So yeah, I think here is definitely some question mark also for me, and also what it means for the data set. Yeah, so, but then we also had like this ICOPS workshop series as well, and there they also address these issues too. And then it is time consuming, and I think many people who are working with phytolith reference images or collections know it's time consuming, and I did this now as part of my PhD, as part of one of the PhD papers. So I actually had the time to do it. But then I also know there's many phytolith researchers that have like a set of images sitting on their computer that doesn't get the time that the set deserves. And so I also wonder what the best way would be to share images in a low barrier way when they're not really part of a main project, but you would like to share them together with the information that's needed to make interpretation of these images. And that brings us to the last part. So that's also my question of scholars need to be able to access these access and also ways to find and use these data sets. And in the past, there has been some efforts done over the last 20 years to actually create these databases. And we have the FeedCore 2.0, which was born in 2011. This is a project from Professor Rosa Maria Albert, which includes digital images and related information of phytoliths from modern plant material, modern soils, paleosoils, and archaeological material. And I think it's a little bit like our modern database for phytoliths. I also often go on there to have a look at phytoliths. And then there's this other project as well, which is the data collected by Professor Deborah Purcell. And I know this was a project that ran from 1997 to 2008, and then also went on until 2015. So I don't know how old the database is exactly. I want to say 2015 that everything was gathered. But yeah, they also have like a website where all the images are. And this is the phytoliths in the Flora of Ecuador project. So that's also a database. And then in 2023, Sigrid Osborne did a master thesis about the British phytolith reference collection. And this then also turned into a website with images. I did check this week and I couldn't open the website anymore. But at some point, I also had a look at all the images. Because that led then to the website that's shown all the plants phytoliths as well of the species that she had been processing. So yeah, that leads us to questioning like what should we do now? And the way I consider the image data set that we built is sort of this piece of the puzzle, but it still has to go to a puzzle. And in that regard, I'm super excited to sort of hear where we are as a community in terms of this data set continuation of where it needs to go towards the future. And what we try to do with this paper is really sort of deliver a subset of images in the most manageable way possible. So yeah, but from here on, I'm also very, very curious, and I would also be very happy to sort of share the data set to wherever it has to go in order for more people to see it and be able to easily use it. So a question that I have is, on the one hand, so how can we improve phytolith images creation, labeling, sharing, and stimulate correct usage? Because during this workflow of this paper, there were quite some things that I did the best thing. I think like I just tried to do what would be best. But I think it would also really be nice to have a sort of maybe similar setup to this document on the Open Phytolith Publishing Guidelines, but then some guidelines on what do we expect as a community, what's in all these images, like what kind of metadata that we want, to also guide some people as well who are interested in doing it themselves. Yeah. And then on the one hand, the other hand is also how can we ensure that the tools that we develop are user-friendly and are easily discoverable. And I think there's also still a lot of discussion, which I am happy to participate in. So thank you for listening, and yeah, very happy to take questions or suggestions, or yeah.
Thank you so much, Rosalie. That was great, I think. I think that you've touched on so many points that all of us who are here today have stumbled upon at some point in their career, or they keep stumbling upon even, now when they sit at the microscope, and especially when they have to publish the results of our investigation. So if anyone has a question, you're more than welcome to put it in the chat if you don't want to speak, or just raise your hand and, well, put your hand up and just ask your question to Rosalie in person, in inverted comma. Yeah, in the meantime, Emma is putting in the chat some of Rosalie's questions for us. So again, if anyone wants to or has some suggestions or an answer for those questions, let's see if we have someone. And otherwise, I'll start. I mean, I've written down like so many points. I mean, not really question, because more or less it's the same points that you raised. But I think that I want to start from one of the last point that you made, which is how we find and how we use this data. So as you said, there is already sort of a lot out there, not only like the databases that you showed, FitCore or the Missouri Lab, or, other visual databases, but there's also loads of information that's, included in papers and, published. So in a certain sense, like the information is there, but it's very fragmented, and sometimes it's on, as you said, like obsolete website, in a sense, it was there and then all of a sudden it's not there anymore, because when we're thinking about images and, well, in general publication of data, we also have to think about the sustainability of the way we published this data. So this is more like another point of reflection for the whole audience to think about how we can, like, sort of, if it's possible to concentrate in one place, maybe not what's already out there, but at least what's going to be created from now on. And there are some databases or some platforms that are already sort of dealing with that. I can think about the Neotoma platform or database for paleoenvironmental data, but there are many other. There's the Global Biodiversity Information Facility and all these type of things. So do you think that could be a way forward, in a sense?
I think so. Something from my perspective that I would really like is if we have like this GBIF database. I don't know if everyone is familiar with it. Maybe we can actually have a look at it as well. Is it still the right screen that's sharing?
Yeah, it's perfect.
So it's this data set that people who are working in herbaria as well, in herbariums, working with modern plant material as well, or occurrences, is basically, you can interrupt me, Carla, if I'm not telling it the right way, because I haven't used it that much. But it's a big collection of several collections from herbariums or from occurrence data as well. So something that I would like to sometimes use it for is if I want to know where fake is occurring, you get like this world map of all the occurrences. And then there's two types of occurrences. So you have either the herbarium specimens, or if people have actually been seeing it somewhere and then they took a picture and then they uploaded it. I think in this table. So here, for example, you see it's like a human observation, for example, data set that it came from, and then some other information as well. So here you can see all the types of observations that you actually have. And I think so my dream would actually be if we could have this for phytoliths as well. And I don't know, of course, if how easy that will work as well, because then I'm also thinking that, of course, for like, it's not just one picture per plant. For like for a specimen, you will have several images as well. And so I also wonder how easy that will still be to manage, because yeah, and then with all the different morphotypes as well. So it's definitely going to look more complex, but it's so, especially for like the herbarium specimens or like the human observations, it's so user-friendly. And I think that from my perspective, we shouldn't try to build something that is where other people have more experience in. So I would really like if, like, sort of from like a phytolith perspective, we could just hook on to people who are very experienced in these databases. But then again, this is also a point where I'm talking about something that what I would like to see happen, but I'm not super familiar with all these things as well. So I don't know if someone would like to add something to that as well.
Anyone has experience working with this type of databases that do you want to share? Or I think that okay, Marco has raised a hand and we have a question in the chat, but I think that just to finish on this topic, I think one advantages of working with this type of platforms is that somehow the information that you put in has to follow certain standards. So that would also cover the issue that you were saying, that what type of metadata do I add to the picture, apart from, the species where it comes from and the name. I mean, what other type of information and what other type of data we can add. And so if we work with something like that, everything has to be standardized. So everyone is basically adding the same information to the images. So, Marco, yes?
Yes.
Yeah.
Let's see if I can get the...
Mm-hmm.
Yes. Thank you so much, Rosalie. It was really, really interesting, and it's enormous work what you're doing. It's really impressive. I think that there is a key problem in all this, and I don't want to be negative. It's actually, I just want to, and I think it's been picked up on both on you, from what you were saying and from what Carla was saying. That first of all is a standardization of the data that needs to be inputted into a system, and I still see a lot of I don't know, maybe it's not the right word, but a lot of anarchy in which we are doing, in the way we're doing this, and it's related to many reasons, but it's very, it's quite chaotic, the information that is there, that we collect, on what we focus and what we make available. And that's the first thing. And the second thing for me is the fact that there are two decisions when we think of a database. One is the decision of what is the software we think would be the best. And I totally agree with you that we need to go for something that maybe has been already developed, and then it's been tested and trained and for similar data, and there is just the need to tweak something and make it for the phytolith. But the biggest side for me is the responsibility of the management of this and the sustainability of the thing, in the sense that for how long it stays where it stays. This is one of the things that Fritz and I discuss a lot with Rosa Maria about the FitCore database, in the sense that, if these databases are based, or in Missouri, are based by where we are working or thing like that, eventually we retire or the funding is cut or whatever happened, and they might disappear. So I think that from my perspective, the core question that as a group of people working on phytolith, we need to ask ourselves is, how can we solve this? What would be the way to solve this? And then all the rest, from my perspective, is a cascading situation in which, yes, there will be a lot of bumps on the road, but it's down to the people to make it work. But the first part is really a question of money and a question of where to put, where all the energy, because this is a lot of work, where all the energy that people are going to put is going to go, and it's going to go to a place where we know that in 20 years' time it's still going to be there. Because also, as you were saying in your presentation, about the names, now some of the names you use are going to change. So somehow that database, not specifically, maybe even the one you upload, but a database should have somebody that goes back and say, oh, okay, eventually this change, or we need to find a solution for these changes so that people are able to drive back, to have a thread to go back. So I think the reason why we don't have it is because there are a lot of unknown in here. And definitely, I think, but the good thing is that we are talking about it. The good thing is that we are thinking how to solve it and what is the problem, and connecting to, that goes back also to what Abraham was saying on the question. Was like how, it was a question for you, Rosalie, where he was saying how can we integrate our photo illustration or whatever there is there available into your database? And this is a kind of a circular thing. My perspective will be we shouldn't. What we should do is to first find a place where we create the database for everybody, and then eventually each one of us with all this luggage can come and put it in. And I think that's why your kind of a discussion and the presentation you gave are important. Thanks.
Thank you, Marco. Yeah, I agree with many of these things. And I think also indeed the question from Abraham, how could this be integrated in your database? It's the key question. And I am very aware that indeed we don't have it yet because it is very time consuming, it's resource consuming as well. And someone should step forward to be able to like, okay, let's do something and let's move forward. And then I hope it sort of becomes this tool that can operate with good rules more or less autonomously, so that people know what to do, that it's very streamlined, that the anarchy of the phytolith research is a bit tamed as well. But yeah, I have been thinking about this as well. And then just like for this paper now, what I try to do is just deliver this package that's a very neutral package that can go in whatever format as a sort of building brick. Because yeah, it's a question of who should do the work. And yeah, that's what I was also, I'm happy that we are discussing it because it's something that I was hoping to get more answers in this this meeting as well. and answering
You
them.
got more question probably.
Yeah.
No, I think at the moment you are the one who is working more on this, so you are the one with the answers. Yeah. Is there anyone else in the audience? Does anyone else comment, an answer for Rosalie, or a question for Rosalie, or for anyone else? I was just I was thinking about what you were saying also. Yeah. I think that, you know, when the people started their own, that like not their own, but their effort on creating the image database, there were like a specific situation also in terms of technology and what was available, you know, and power, you know, like storing power for like big data sets and things like this. Now things are changing. As we were saying, there are loads of not loads, but you know, there are places where all this is being taken into consideration, and that there are repository, like larger repository, that are not attached to a single person and an institution, but they are like an international effort, let's say, made by international institution or groups like the GB, for example, but many others. So I guess probably that's one way we want to try and go to, rather than, again, as you both were saying, creating something new again, which is going to be a lot of effort, and you know, and it's not needed because things are already out there. So no question, just praise. Thanks, Agnes. Well,
Question from Helmut.
yeah, Velmut. Yeah, a bit more about the Lightroom program. Yeah.
Yeah. Okay, let's see if I can share my screen again. Okay. So now we're in Adobe Lightroom. It's this icon here. So I decided to use Adobe Lightroom. It's a software, Helmut, for photographers who have to deal with their image library. So it's for them a way to easily have different albums. You can also share albums with other people. You can add keywords. It's also a software where you can actually like modify your images, like do some like light settings and all these things, but it's something that for our work is not needed. And so now you can already see the problem with that. It's like something that you need to renew. So a couple of weeks ago, it's like with these 1-year renewals. So I still have to do it for this year. And now I'm already disabled by many things. So my search is disabled. I cannot do my keywords anymore. And so I think that's the downside of it. But what is really nice is that you do have a sort of big folder, and normally you are able to search on keywords. I don't know if I can say. So here, for example, these are all my keywords that I added to the images. So which corresponds to the keywords in the paper then. And then normally we can click on it. So you can click on it, and then I get all these images together in this case, it's like a morphotype that's being found in nettle, which is a hare with a bulbous head. And so here, for example, you can see that it's here in the background, and here is another type of hare. So then it is the acicular bulbous is this one, and then the trichome number 3 is then this one. So yeah. Let's see if I can. Oops. So it's a nice way of actually going through your images as well. So even just for yourself as well, like if you don't want to publish it, but just have a good overview, because I know sometimes with my Phytolith images that I know I've seen something somewhere, but I can't really remember where the image is. And so then it takes me a long time to actually look for something on my computer. And in that regard, it's a very nice way of photo organizing. But it is not a scientific tool. It's a very commercial tool for photographers. Does that answer the question?
Yeah, I think so. Yeah, in the chat there were like a couple of suggestion. ImageJ as an alternative to Lightroom. ImageJ is like image processing software rather than image library, or I don't know if that can be both. And Javi was saying Darktable works in a similar way to Lightroom, but it's open source. And
Okay.
yeah, I didn't know about that. That's definitely something to explore. Darktable.
Darktable. It's also new for me, so it's also something that I will explore as well. Thank you.
So yeah, also like there are lots of I think, as I was saying, there are lots of tools and available out there now that we can explore. Marco is a great admirer of Lightroom, so
Yeah, well, I went through a lot of going through different software for fundamentally for fieldwork images mostly, and I have 50,000 images, and you want a software that is, quick and easy to deal with. And I do agree with you, Rosalie, that Lightroom is fundamentally one of the best because it has, as you were saying, the two things. It's a database that you can search and you can structure it the way you want in a sense. But it also has a big advantage for the type of images we use, which it keeps the original. Whenever you do a modification on the image, for example, you adjust the contrast or the sharpness or whatever you do, it still keeps the original image without modification. So that means, contrary to, for instance, all the software that saves in JPEG or other type of, and that's very good because it means that your original data is always there. You can play with it the way you want, but your original data is always there. I agree. It's a problem that is a payment, and now they move from paying to buy the software to subscription, so you have to pay regularly. That's quite a pain. But indeed, at least me, I haven't found.
It looks like Darktable works in the same way with raw data, and it says that the image is only converted during export if you do export it. So
okay,
have a look at it.
Yes, we will have a look at it because I think it's not only when you export that it modifies the images, it's also when you do the modification and save them in the system. That's many software. What they do is they modify it.
This one says that it is only the full image is only converted during export. Raw images kept. So I mean, we can have a look. It's definitely a good
of course.
suggestion. Thank Javi.
Yes, thank Javi.
That's good. Any other comments, questions, suggestions for Rosalie, for ICOPS, or for anyone else? Otherwise, I guess we are at our 1 hour exactly, like perfect timing. So yeah, so just remind you, as Emma has put in the chat, that next speaker will be Francesca D'Agostini. The host will be Emma. Francesca is now a researcher in Kew Garden, and she will speak about how to start implementing open publishing. If you're interested in that, there's the Zoom registration link in the chat. And also, as I was saying, if you would like us to organize a specific seminar on a specific topic that it's of interest of you, or if you know someone who's really great at some aspect of open science with phytoliths, or I mean, not necessarily. It can be also someone who works with other type of data, but that uses a method that can be maybe applied by us working on phytolith. That as well is very welcome to come and give a seminar. So I guess no more comments, no more questions. So I thank everyone again for coming to this seminar. I hope to see as many of you as possible to the next one and to the following as well. And that's it. Have a great evening or day, and then from tomorrow, a great weekend to all of you.